Good morning. My name is Paez. I'm a technical marketing manager here at Aldevron. I wanted to briefly take a moment to thank you for your time and for your interest in attending our talk. We're going to discuss our next generation plasma technology called Nanoplasmid in the context of non-viral applications, and hopefully I can cover some of the most recent reports and answer some of your questions. So Aldevron at a glance. Some of my colleagues like to say we're a central dogma manufacturer, and that we manufacture plasmid DNA, mRNA, and proteins like CRISPR-Cas9 into nucleases. We've been in the cell and gene therapy field for 25 years. I've had the pleasure, honor, and distinction to serve some clients who've been doing some of the most groundbreaking work within the field. We have the largest GMP-grade plasmid facility in the world. And as a company, we're deeply devoted to our clients, ensuring that their therapies work and that we serve them at the best possible level because we think that they're doing groundbreaking work. So looking at our manufacturing footprint, we have our breakthrough campus in Fargo, North Dakota, which is the largest GMP plasma manufacturing facility in the world, where we make GMP source and GMP manufacturing of plasmid DNA, RNA, and proteins. We also have an advanced campus in Fargo, North Dakota, where we make research-grade DNA plasmids, research-grade RNA, and we do some research and development there. We also have our Madison campus, which is ISO 13485 certified. They do custom process development for proteins and manufacturing of CRISPR-Cas9 endonucleases or anything custom that you may need. And then finally, last but not least, we have our Lincoln campus, which makes the research-grade nanoplasmid vector and the proprietary Reviver host cell strain line, along with our batch-fed hypergrow manufacturing platform for fermentation. So before we start this talk, I'd like to start with this figure here in orange that is pulled from a review article that was published in Molecular Therapy and Nucleic Acids by Dr. Jim Williams, who's the inventor of Nanoplasmid, and myself. And what this figure is showing here is a timeline of the major approvals and clinical trials within the cell and gene therapy space, and a timeline of when puck plasmids were initially developed, which is in the 1980s, relating to their structure and their function of their use within the field. So the first thing with the structure is puck plasmids typically have what's called a bacterial selection marker, as you may know, of ampicillin or kanamycin, or CAN for short. And what was seen is that early on with these puck plasmids, they saw that in the wastewaters of laboratories, you would see that these plasmids were ending up in other organisms and were contaminating. And so this led to the idea or the creation and understanding of horizontal gene transfer. And so we saw that. And then relating to the same structure, as folks continued to use these puck plasmids to manufacture AAV, you started to see reports of vector-mediated backbone antibiotic resistance transfer with AAV plasmids that had been packaged with that, because what would occur is sometimes reverse packaging of the ITRs would occur, and instead of packaging your transgene, you package the backbone of the plasmid. And so finally, with function, you started to see that there was vector backbone-mediated transgene silencing, there was cellular-mediated, backbone-mediated cellular perturbation, cellular toxicity, and all sorts of things that occurred after transfection and use of these puck plasmids. And in the paper, we make the argument that puck plasmids were initially designed for cloning. They were not necessarily designed for cell and gene therapy. They work, but there was room for improvement. So the area for improvement, we believe, is the bacterial region of the backbone. There were sentinel papers that were published discussing why there was transgene silencing and why there was post-transfection toxicity. And basically what they found out is that the size of the backbone was very important. And it wasn't just the size, what was in the backbone is what was a problem. The fact that there were bacterial coating regions within the backbone of the plasmid were an issue. And that was killing cells after transfection. So these are the thoughts that led to the creation of the nanoplasmid vector. The nanoplasmid vector has a very small backbone. 500 base pairs. In addition to having a 500 pair base backbone, it has a selection system called RNA out, which uses sucrose. And furthermore, it's R6K strain dependent, and I'll tell you why that's important in a later slide. But the general idea is that this next generation plasmid has high transgene expression, diminishes cargo size restrictions, reduces transgene silencing, and has a ton of manufacturing advantages in the downstream for our customers because you kill less cells and you have more to work with at the end. So let's talk about the RNA out selection system. The way it works is fairly simple. The proprietary host strain called Reviver expresses something called SAC-B. This gene SAC-B codes for this protein called levain-sucrase. When we're culturing this E. coli strain, we culture it in a sucrose medium. When this levain-sucrase is expressed by the SAC-B gene that the bacterial hosts express normally, levain-sucrase reacts with sucrose and concatemerizes and breaks the E. coli strains, leading to death. Any cell that is competently taken in the nanoplasmid will express this RNA out sequence. And RNA out inhibits the translation of the SAC-B gene by mRNA, leading to survival. And that's how we select it. It's very safe. It just uses sucrose. It's easily scalable. Speaking about scale, it's easily manufactured within Reviver host strain. Our fermentation process can yield 1 to 2 grams per liter easily, sometimes more. So it's highly scalable. So if we compare it to other DNA technologies in the market, I would say that it's kind of a progression. So if you start with typical plasmids, their bacterial region size is greater than 1.5 kb. And that causes, what's been reported in the literature, is increased transfection toxicity, increased transgene silencing, low transgene expression. The selection, as we know and we discuss, is antibiotic. It is high yielding, and yes, it is scalable. But in terms of another layer of safety, it's promiscuous. Puck plasmids are typically promiscuous in that they will go into other bacterial strains in the environment or even in a patient if they make it into a patient. And so that does provide some layer of danger, and that's something that we wanted to mitigate. So the field seemed to move on to mini circles next. They said, okay, well, we're going to cut out the bacterial region. We're going to make the backbone smaller. And they did gain some performance benefits. They did have reduced transfection toxicity, reduced transgene silencing. It was still antibiotic resistance selection, but through a series of recombinases, they were able to cut out that backbone. But the problem with that is that they made the manufacturing yield very low and the scalability of the manufacturing very complex. And so a lot of folks, while they like the performance of mini circles, it became somewhat financially unfeasible to scale. This is where Nanoplasmid comes in. It has a very small backbone. This size is backed by science. This is the Goldilocks spot. We know this. And it shows reduced transfection toxicity, reduced transgene silencing, high transgene expression. It uses a sucrose system, that RNA out that we just discussed. It's high yielding, it's highly scalable from research grade all the way to GMP. And finally, it's R6K strain dependent, meaning it's not as promiscuous as typical puck plasmids. So let's talk about some of the data that's been published by academic researchers and others. In this paper, they were electroporating and delivering either a standard puck plasmid in a GWiz platform or a nanoplasmid in vivo and in vitro into cardiomyocytes. And what basically we saw is that in red here is nanoplasmid, and this is the gene expression. Nanoplasmid had significant gene expression at day 1 after electroporation and day 2 as compared to conventional plasmids, which are in blue. Not only did it outperform in terms of gene expression, but it also did so at a lower voltage, so 90 versus 120. So now let's talk about manufacturing in terms of using it for mRNA. Nanoplasmid, it uses the Reviver host cell strain line to manufacture. Reviver is very good at handling structural DNA elements like poly A tails, which are seen if folks decide to put the poly A tail at the time of linearization, right? And so what we did here is we compared a traditional puck plasmid versus a nanoplasmid in an A99 vector. And then we've compared, again, the same puck plasmid versus a nanoplasmid in a P60 vector. And what we found is that we were able to yield 5 to at least twofold over a traditional puck plasmid vector while retaining the poly-A tail repeat integrity. So now let's talk a little bit about where the rubber meets the road. People are starting to report how they're using nanoplasmid in some of their non-viral applications. And in this report, this is Genentech, where they made CD8 CAR T cells, and they were using an HDR method to knock in TCRs. And they used our Aldevron SpyFi-Cas9 and an RMP, well, complex, right, to knock in these genes. And they compared three things. They compared to 3 donor templates. First, they compared using a linear double-stranded DNA as a knock-in template for CRISPR HDR, then a conventional puck plasmid, and then a nanoplasmid. And what they found is, in terms of knock-in efficiency and total number of edited cells, nanoplasmid had a higher frequency. Much higher frequency, in fact. It was twice the number of cells as compared to linear DNA and 3 times as compared to puck plasmids. Another group from Editas Medicine developed another CRISPR method, and they called this SLEEK. This is a very special enrichment method. SLEEK stands for Selection by Essential Gene and Exon Knock-in. And it basically results in the death of cells performing non-homologous end joining. And so the way this works is you have a cell of interest, you have a CRISPR RNP targeting an essential gene, something like GAPDH, something cells can't live without, right? And then you have your donor DNA target containing your transgene cargo. You electroporate, and then you let the cells recover for 7 days. What ends up happening is you have 3 outcomes. You either have unedited cells, they didn't get the RNP, they don't have any cuts to the transgenes or the essential genes. You have cells that die from non-homologous end joining because you're cutting the essential proteins like GAPDH. They can't survive. So if they're not getting the essential gene back in addition to the transgene, they're gonna die. Or you get cells that go through homology-directed repair. And so they're given back the essential gene for normal cellular function. And so what they found is they could knock in 90% efficiency with either a nanoplasmid or Aldevron's linear double-stranded DNA. And so the other thing they found is that this expression of these genes were tunable, depending on what gene you targeted, whether you go for GAPDH, KIF11, or TBP, which is responsible for transcription. And so here you can see on a histogram flow cytometry of how tunable it is. So the highest expression being GAPDH, followed by KIF11, and so on and so forth. And finally, in the paper, they showed that you can use SLEEK to insert up to 4 transgene cargoes simultaneously into one cell. Here's another example of a group out of Stanford using our nanoplasmid. They went a little bit of a different route using CRISPR. They did something called homology-independent targeted insertion. So it's not necessarily HDR, because this allows for knock-ins from both non-dividing and dividing cells, right? Because in HDR, you need the sister chromatids in order to have a template and have homology, right? So what they did is they grabbed the nanoplasmid and they knocked it in to this, they used the GD2 CAR T cell, which is particularly relevant in the case of neuroblastoma. It's a popular target right now, as many of you know. And they knocked that into the track locus, and they used the dihydrofolate reductase enzyme, and they gave it back within this nanoplasmid because they used an enrichment process through methotrexate. Methotrexate you can rescue cells that are being treated by methotrexate, a common chemotherapeutic, by giving them back this gene because it allows for nucleotide synthesis to come back into the cell and survival. And so they call this enrichment process called cement, so CRISPR enrichment process. And so what they did is very similar to the previous paper. They grabbed T cells, they activated them, they electroporated them using a nanoplasmid and an RMP, and then they started optimizing with their methotrexate selection. And so any cell that successfully took in this plasmid would confer resistance to methotrexate selection and thereby survive. And they were getting CRISPR knock-in efficiencies of about 80% using clinically relevant targets, and their protocol lasted about 14 days to have very functionally active T cells. They also went ahead and they did it with donor T cells from patients who were undergoing chemotherapies and so on and so forth from other clinical trials to see what it would look like in the context of maybe a patient who is not necessarily a healthy donor. And they saw similar results. So it was a very robust system using our nanoplasmid. So nanoplasmid's not just been used for CRISPR, it's also been used for transposon applications. We've seen it used with Piggyback, TC Buster, and Sleeping Beauty. And what we find is that it improves transposition rates, it lowers transfection toxicity, and again, we address some of those regulatory concerns because we don't have the antibiotic resistance marker and no bacterial-coding proteins. And we're really not seeing any changes to the sequence or composition of the integrated transposon. So one of the case studies that I want to share with you today is Paez Therapeutics, where they published a report that you guys can look up, where they used their BCMA CAR T cell trial. And they found that in their manufacturing process, they were able to increase transposition frequency, they were able to shorten their timelines, they had good expansion in the clinic, and they had a really great clinical response, which I'm going to show you in the next slide. In the BCMA 101 trial, they had a small cohort where they gave patients either CAR T cells that were manufactured with a normal conventional puck plasmid, as they usually used, or CAR T cells that were manufactured with a nanoplasmid. And what they found is that we had a significantly higher overall response rate as compared to patients that received a traditional puck plasmid manufactured CAR T cell. So 100 versus 0 in terms of a very good partial response or complete response, and 100 versus 50 or 67 if you're looking at the International Myeloma Working Group diagnostic criteria. So nanoplasmid safety profile was also preserved. There was no cytokine release syndrome observed with either of these products in these patients. And they reported this, and it appears that these CAR T cells are still well and good working within these patients. So we're looking to hear more about this in the future. So let's talk about nanoplasmid in terms of clinical applications. How many clinical trials are we in? What we can say right now is that we're in about 13. It's being used in about 13, 8 completed phase 1s, 2 recruiting or ongoing phase 1s, 1 ongoing phase 2, and 2 completed phase 2s. And here's some of the select clinical trials that we can publicly speak about. Paez Therapeutics is the one I already spoke about here. This one here is very interesting. This is enGene. This is their therapy called EG70. They use a nanoplasmid that encodes RIG-I and IL-12P40, and it is intravesicularly injected infused into the bladder of cancer patients. And it's a gene therapy in order to stimulate an immune response within these patients who are being treated with either BCG or are refractory to BCG in non-muscle invasive bladder cancer. Early interim reports show that they had a 67% response rate, and there was no severe adverse events or SEs that were any more or any less than what you see with BCG vaccine treatment. And so finally, just for a quick conclusion, we talked about non-viral gene therapies, how we can increase transgene expressions, lower transfection toxicities. We can help with end product production in terms of non-viral gene therapies, whether we're talking CRISPR or transposon. We can help with plasmid template in terms of mRNA vector production. We're also doing a little bit of work to address our viral vector folks. We have some evidence that we're not ready to show just yet that we can help manufacture AAV and lenti, but as you could imagine, the safety profile of it is very attractive for AAV and lenti, and hopefully maybe next time I'm up here I can show you a little bit about that. And so in terms of what does this look like? If you want this in your pipeline, what is this going to look like? You can come to us either with your traditional plasmid and we will cut and paste it or retrofit it, as we say, and go ahead and start manufacturing for you. Or you can give us a gene of interest and we'll do all the cloning for you and manufacture for you. We support from research and development and discovery, so RG all the way to GMP and commercialization. And we have a little intermediary here, which is called GMP source. It's got all the quality of GMP, but not necessarily the paperwork. So it's good cost savings for some of the folks who really need to make sure they have high quality. And so with that, I will take any questions, and thank you for your time.
Just a quick one. So do you need your whole cell line to make the plasmid, nanoplasmid, or we can use any other?
No, great question. You need to use the Reviver host cell strain line because the origin of replication is R6K strain dependent, and so it could not go into a regular DH5 alpha cell line.
Okay.
Correct.
Thank you.
Sure. Yeah.
Hi.
Hi.
Great talk. Some really great results. So I thought I liked your timeline, so I'll ask you what's going to happen in the next 25 years of plasmid technology?
Ooh, I'm sweating. Next 25 years, that's a big question.
It's 25 on the slide, so I thought.
So 25 is because we've been in the cell and gene therapy field for 25 years. What's going to happen in the next 25? I hope that all of your therapies and dreams come true. That's what I hope. But you know, all kidding aside, I think that as these therapies get out and become more commercially available and approved, we're really going to have to start talking about safety. And I think that using next generation vectors that provide safety, but also make the feasibility of manufacturing better for our clients, I think that's going to be a big deal, right? Money's a thing, and this can save you money, we believe, and give you the performance that you need. You know, it's like turning on the lights at full blast versus halfway. That's what we think.
Maybe picking up on that question, is
Sure.
Is sort of the T-cell ex vivo cell therapy the sort of the biggest area for, at the moment, activity, or are you seeing others?
No, we're seeing a lot. So a lot of CAR T-cell is very popular as evidenced by this conference, but we're seeing people use it as a direct vector. We're seeing people use it to make mRNA. We're seeing it, we're just seeing it in very, very, very diverse applications. For viral applications, there are reports where folks just used our RNA out, which is half of the secret sauce of Nanoplasmid, and they were able to make serum-free lentivirus, and they were able to use 50% less plasmid and gain twofold viral expression or viral titers with lentivirus, and they just got half the secret sauce. And so we're rapidly working to show you what can be done for viral vectors, but I can tell you that data I can't share with you, but from clients who are using it, they're very happy with it, and they're adopting it very much in the viral vector world. And so it's pleiotropic in its applications right now.
Any other questions? All right, if not, let's thank Patrick once again.
Thank you.